Good morning students. Today we will discuss in this session in the fourth unit, that is recombinant insulin process. Yes, in the fourth unit what are our main topics includes here? That is simply we already discussed that hybridoma technology concept and their applications is over, and the next one is stem cells types and their uses. Okay. In this fourth unit, the first topic is includes in vitro fertilization and embryo transfer. And let us discuss in this session the remaining topic which is continuous in the last class, that is stem cells types and their applications or the uses of stem cells. Okay, here stem cells. Which cell can the blastocyst cells which can modified into another types of tissues. Okay, they have the capability to producing of the whole organism, including of placenta. Such type of stem cells are called totipotent stem cells, and next one is pluripotent stem cells. Except the placenta, remaining all types of all kinds of tissues are formed by the cells. Those are simply called here pluripotent stem cells. And next one is that is multipotent stem cells. Here these we give a simple examples to in this multipotent stem cells, that is hematopoietic stem cells. Here they are simply produces these all type of blood tube cells. Blood is a connective tissue, RBC, WBC and platelets, so and so on. And next last type of the stem cell, that is simply unipotent stem cells. These are derived differentiated from the multipotent stem cells. Yes, just I am recapitulated to the last class, ma. And this the name is indicates as a single type of cell or tissues are produced. Yes, here the best example is mast tissues. The cells only capable to synthesis of or the modifying of the only mast tissues. And the remaining two types are, that is an embryonic stem cells and adult stem cells. Right? Embryonic stem cells, they are differentiated into the organs. Which organs are needed to this same organism, they are synthesized or they are produced in the process of in vitro conditions. The stem cells are used here to treat with these different type of diseases or diagnosis. Okay, these stem cells are mostly applicable. So what are the applications and what are the uses of these stem cells? Okay, the different type of diseases, those are prevented by using of these stem cells. Here first of all we watch here, that is the diabetes, and next one is the Parkinson's diseases and Alzheimer's, and next one is the cancer and some relative diseases, brain relative diseases, and some are the relatives to the spinal cord. Okay, these both the spinal and brain, both the central nervous system, a complete system. Okay, the type of diseases are prevented by using of these stem cell therapies. Stem cell therapy. Let us be discussing how the stem cells are used to prevent these diabetes diseases. Let us discuss in a few more points in this class, that is the application or the uses of stem cells. Yeah, these uses of stem cells. The first one is to prevent the diabetes. Prevented the diabetes disease. Diabetes using these stem cells. Right now, here diabetes. How we define this disease? Nowadays, this disease is spread as a code. Okay, and 100% members or the population, approximately the 75 or persons are have these diabetes. Okay, how it is occurs? Let us be discussing some more points too about these diabetes. The levels of blood sugars. Okay, either the increasing or decreasing of the levels of blood sugars. Okay, this disease is caused due to the deficiency of the hormone. That hormone is insulin. Which type of hormone? Deficiency of hormone. Hormone in the blood levels, the hormone is here insulin. This insulin is produced by the pancreas. Pancreas is a mixture gland or a mixed gland. Right now? Mixed gland. This pancreas, the structure is located in this our digestive system. Here the U shape. Okay. Here the leaf-like structure of the pancreas is present. This is the pancreas. In the pancreas, we are observing of the inside the TS of pancreas. We are find out so many highlights of Langerhans. These highlights of Langerhans can synthesis or producing the insulin. These highlights of Langerhans can be producing these two types of cells, that is alpha and beta, and so many other type of cells is there. Especially these beta cells have the capability to producing this insulin. Right? What is our main hormone is insulin. This hormone insulin is released from these pancreatic islets of the Langerhans, and it is involved in the bloodstream. Alright? And it is slowly reached in our total body. Okay, each and every cell is can capturing of these glucose levels. Then only they can producing some energy, ATPs, right? And here the in the diabetic patients, what is occurs? In the diabetic patients, the blood sugar levels are enhanced and the glucose levels will be either increased or decreased. If the glucose levels are increased, these high amounts of glucose will excreted by their urine. From their urine, excreted excess of glucose from urine. This is simply regarded as diabetes mellitus. Diabetes mellitus. Diabetes mellitus. The excess of amounts of glucose will be excreted from the urine. The patient condition here. The patient condition is like this. Then only we treated or we diagnosis their urine samples and their blood samples, and we are confirmed that the person is suffered with this sugar or the diabetes. Alright. And next here, nowadays the patients can suffer with these diabetes. Doctors can prescribe them if their level will be increases to normally treated with these tablets or the medicine or the drugs. Okay, if it is not possible, they can directly write some injection. The glucose, okay, hypoglycemic conditions of these type of glucose can slowly injected some injections, and they are directly given themselves only. The patients directly given by themselves by these injections now. Right? That's why the persons here suffered with this taking of number of injections into their body. That's why some scientists do this so many experiments. In our olden days, we are extracted these insulin from the animals, right? The animals are here. The first animal is pig, and next slowly we can synthesize this insulin from the cattle also. Okay, here we using the animals to making of these insulin hormone in the high doses. Alright, that's why nowadays there are running therapy. The running therapy is to transplantation of the pancreatic gland. Right? when the patient is severely suffered with this diabetes, okay, if we have an economical support, the doctor suggests to him to transplantation of this pancreatic gland. That's why here the pancreatic gland, okay, entire gland will be retransplanted. But some situations it is stable, and some conditions it is heavy, and the immunity will be not supported to the person. Okay, so many other causes it is taking more and more expensive, and it is also take more time. That's why simply the person here what the doctors are suggest, not only the pancreatic gland fully transferred, sometimes the highlight of Langerhans also be transplanted by the patient. That's why here what occurs, they are synthesized. The highlights of Langerhans also be synthesized, and these are only transposed to these patient. Right now, this is about the simple using of the stem cells and to recure or prevent the disease, that is the simple diabetes. Okay, here remaining instead of total gland transplantation, the scientists are used only the highlights of Langerhans. Just they are separated the highlight of Langerhans and to implemented into or implantation into the inside of the pancreatic specific region. Okay, and next their cells are ready to secreting this insulin or the beta cells. Now these beta cells are slowly secreting the insulin, but it is taking a long and long process. Okay, sometimes it is also give some the negative results. Not always in positive results we can't get. Sometimes we also get some negative results. And one more is that the role of stem cells to give a treatment in the Parkinson diseases. Another disease, the Parkinson diseases. What are the symptoms of Parkinson disease? The neurotransmitter, okay, the nerve cells in our brain, the loss of memory. It is also one of the type of loss of memory. Alzheimer's is also one of the loss of memory type. The symptoms are head, legs, and their hands are shivering involuntarily. Right, ma'am? Here another disease can also be treated with these stem cells. That is the Parkinson disease. Parkinson's disease. Alright. In this disease also using of these uses of stem cells to prevent of this disease. Okay. What is first of all we learn to know about what is the symptoms of this Parkinson disease. The symptoms of the patient is head, legs, hands or shivering. Right now, which means shaking, shivering, involuntary. They are not control the person. Okay, the movements are not under controlling of the person. Suddenly they are high shiver. Even in the resting condition, not only do any works, even in the resting conditions also. The main reason of the Parkinson disease is the non-secretions of the neurotransmitters. Okay, non-secretions of the neurotransmitters that are linked with the nerve junctions. The neurotransmitter here releasing of a special type of chemical. The chemical name is here dopamine. This dopamine, when this dopamine is secretes more and more and it deposits in the nigra, the region of our brain, okay, nigra region of our brain regions. It is we are having in the two, okay. Here At the Niagara region, the depositions of more and more dopamine combined deposits in the Niagara region, the person is lost their memory. And also the condition, the voluntary muscle conditions is also not under controlling of our nervous system. That's why always they are shivering. Okay, and here the doctors have suggested that the drug is L-dopa. Right now, here the drug is L-dopa or levodopa. Dopamine is an secretion, chemical secretion, which is more and more essential to this our disease. Right now, and these stem cells of rat and monkey. Okay, how this Parkinson disease? The stem cells of rat and monkey taken in this method and to cure the disease or prevented this Parkinson disease. Okay. Yeah, I hope that you people understood my way. Okay, ma'am. These two diseases are prevented by the using of these stem cells. Moreover, Alzheimer's and cancers and some brain diseases and some spinal cord related diseases are also treated with the stem cells. And next topic is recombinant insulin. How the RNA technology used to synthesis of the insulin in this diabetes. Okay, the recombinant insulin, how it is produced. Okay, ma'am. In this session, we will discuss the recombinant insulin. Insulin is the first human gene product. Insulin is the first human gene product. Right now, this is a human gene product. First, actually in our olden days also we have extracted this insulin from the pigs. So nowadays, the manufacturing of using RNA technology. Now this first human gene product is manufactured by what, ma'am? Using of RNA technology. RNA technology. What it occurs? Here simply we are first same as usual. We will give some introduction about the insulin. Insulin is produced by the beta cells of islets of Langerhans. Right? These Langerhans are situated in the pancreatic gland. Pancreatic gland is act as a mixed gland, exocrine gland and endocrine gland parts. Both are not. Yeah, it is a hypoglycemic hormone. The insulin is a hypoglycemic hormone reducing the blood sugar glucose levels. Reducing the blood glucose level. Right. Hypoglycemic condition. Hypo means reducing. Hyper means increasing. Right now. And here the deficiency of insulin leads to diabetes mellitus. Just now we were discussing about this. The deficiency of the insulin, it leads into diabetes mellitus, and excess of the proteins or these glucose levels are excreted from the urine. The patient always suffered with the frequently he went to the washrooms and he always feels thirsty and always he feels angry. Right now, and some persons are always their inner feels like an phobia, fear. Okay, so many symptoms is there to identify these patients which are who is suffered with this diabetes. And next one here, the prior to development of these Himalayan. Okay, the first here how the insulin is developed in using of this RDNA technology. The prior to the developing of this insulin, especially in human insulin, right? That's why this is simply regarded here in the RDNA technology, human insulin, both are including and joined with given a name humulin, right? Ma? HU humulin, human insulin, humulin product, right? The diabetic protein can be dependent upon the animal insulin, and its insulin is extracted from the pancreatic islets of the scattered cattles and the pig. Right, already we discussed now. The highlights of Langerhans extracted from these cattles or the pig, and the highlights of Langerhans can screened in vitro conditions, and we do some highly precautions are also be taken by the scientists, and they are arranged and they can ready to producing an high amounts of insulin, right? And let us we discuss here the currently this insulin is made to using of the Escherichia coli bacteria. Okay, the simple synthesis of the Escherichia coli bacteria. Okay, and they are making the RDNA technologies. Alright, how the process are including here? Let us see. Right, the bacteria name is here E. coli, Escherichia coli. Okay, in the Escherichia coli, this insulin is human insulin. Insulin is made up of with the 51 amino acids. It is a protein, a glycoprotein. It is made up of with 51 amino acids. In this, it containing two oligosaccharide chains. Chain in one, a chain is consist of the 21 amino acids, and next one is here. Next, another chain, that is second chain, that is simply beta chain, is consist of 30 amino acids. Okay, let us keep in our mind. Okay, this one. And next, the antibacterial role, which is insulin, was first discovered by these antibacterial roles. The scientist, who is the scientist first discovering of this insulin product. Okay, his name is Sir Banting. Is somehow F.G. Banting. And another scientist name is Eli Lilly. In the these both are belongings to the USA, and these are first discovered by this insulin in this RDNA process. Alright, ma'am. And these are started to produce in the human insulin from genetically engineered bacteria. Human insulin is made up of 51 amino acids. They are arranged in the two polypeptide chains. That are okay. Two polypeptide chains. This one chain is containing 21 amino acids, and another one chain is containing the 30 amino acids. Okay, and these are both are linked together by the disulfide linkages. Both are linked by the disulfide linkages. Right? Disulfide linkages. These both are linked with by the disulfide linkages. Okay, both two polypeptide chains. Let us draw the diagram, which is roughly diagram which are helpful to easy. Easy to learn now, right? The first one is here we taking the both sizes, right? A chain and B chain. Here both chains, and we are cutting into in a 18 oligosaccharide cell. The first of all we take in a short, and the next one is a little longer. Okay, both the chains are inserted into the plasmids. The plasmid now it will become a DNA. Okay, and next this plasmid is slowly inserted into the E. coli bacteria. Now the bacteria is production, right, starts their production, cell production, and these coming out of these long one and short one, both are fused, right now, both are fused to form a long chain, the complete double chain process. And the single one, here the A chain and here the B chain. Both chain particles are separated and those are inserted into the plasmid vectors, and now we are getting an RNAs. These RNAs are inserted into the E. coli bacteria, and E. coli bacteria now it get producing more and more copies of cells, and these cells are separated and these are cleaved. These are cleaved by a special treatment, that is cyanobromide. Cyanobromide, right now. Let us be through the simple diagrams which are used in this, right? The first here this is an plasmid. Here we using a destruction engines, and this is the okay. Let's use imagine this is the first A chain. A chain amino acids. Here we using restriction enzyme. This restriction enzyme useful to cut it in a proper manner. Okay, right. And next here A chain also be cut it in a proper manner, and these both are fused to form here and make it in. It is a now just like simply we are connecting up these both, right? This is an special. Now this is an RDA. Excuse me. We are using DNA ligase, which is helpful to joining with this ligase. And now this RDNA is slowly insertion into the E. coli. This is the E. coli bacteria. In the E. coli bacteria, we are insertions. Now it is ready to producing and a chain now. The chain is containing amino acid sequences, right? These are here the occurring of the lysis of cells. Lysis of cell, and you know producing here these. When we are getting these amino acid sequences are cleaved with cyanide, right? That is cyanogen bromide, and we are cleared with the cyanogen bromide. And next one is propagation, and what is one the purification, right? The purification process is also going on, and next we are getting a B chain from this side. Okay, here where we are using either A chain or B chain, we are getting here the A chain. And next here another one Here also be the B chain, and here a restriction enzyme. We use these plasmids. This is the B chain, and next here we are cutting into in this side, and both are fused to form with using of DNA ligase. Maybe appears or not? DNA ligase. DNA ligase. We used it and joining of these both plasmid and the chain system, and now forming as it is in a rDNA. This rDNA is insertions into the E. coli, insertion into the E. coli bacteria. Okay, the lysis of cell is occurs inside, and now it is produced the small chain, that is a B chain. The B chain can synthesis or produced here. The B chain is a synthesis here. Here also be same. This chain is treated with the cyanogen bromide and purification is occurs. Then next we are taking out of this process and this chain, and both these and these both chains are added to form here the double chain. The double chain of insulin is ready to produce here. This is our final product. This final product is produced when we are using Escherichia coli bacteria in the laboratory or in vitro conditions. How much we are getting in a week? In a week we are getting 1 into 10 to the power of 5 of insulin copies are produced or getting within a week. Within a week we are getting this type of copies of insulin can produced by using of this rDNA technology. That's why the insulin is provided where the patient number if is increases, whatever we need in these treatments. Okay, this is about the recombinant